Thank you for watching this pre-recorded short version of our talk at the Great Lakes Bioinformatics Conference 2023. My name is Yu-Chiao Chiu. I'm an assistant professor of medicine at the UPMC Human Cancer Center at the University of Pittsburgh. It is my great pleasure to make this presentation on behalf of our collaboration teams at the University of Texas Health at San Antonio and the University of Pittsburgh. Our study introduces DepLink, which is a web-based R-Shiny app to systematically integrate genetic and pharmacologic dependency maps of cancer. With the advances in high-throughput technologies, large-scale genetic and pharmacologic dependency maps, also known as the DEP maps, have been generated. These projects use genome-wide CRISPR loss-of-function screens to screen through more than 17,000 genes across more than 1,000 pan-cancer cell lines, and also utilized high-throughput drug screens to screen through almost 5,000 small molecules across pan-cancer cell lines. Furthermore, the LINKS project has generated the molecular signatures of 20,000 compounds, which measure the changes in gene expression profiles induced by drug treatments. These data resources have the potential to significantly advance precision oncology by identifying and targeting the Achilles heel of cancer. However, user-friendly software is needed to link these heterogeneous data sets in order to investigate similar perturbation mechanisms among genes and drugs. Specifically, our goal is to match gene knockouts and drug treatments which induce similar effect on cell viability or similar effect on gene expression profiles. Our goal is to answer two fundamental questions. The first is, which drugs are potential surrogates to a gene knockout? And the second question is, which genes are potential targets or mechanism of action of a drug? Our tool has four main modules to analyze similar effects on cell viability induced by a gene knockout and the drug treatments, the changes in gene expression profiles induced by a drug treatment, and to study biochemical features of a new drug. Module 1 allows users to input a gene and search for drug perturbations that achieve similar inhibitory effect on cell viability. Module 2 queries a short list of genes trying to identify a drug that perturbs these genes simultaneously. Module 3 allows users to input a known drug and search for genetic perturbations with similar inhibitory effect on cell viability. Module 4 is designed for novel compounds. Users can input the SMILES codes of a novel compound. The module converts the code to biochemical fingerprints and then searches for similar drugs among 5,000 compounds in our database. Here are some screenshots of the homepage. The homepage provides summary of all data sets integrated by our tool, which include the DEP maps of the Broad Institute, the Sanger Institute, the drug screen of the PRISM and the GDSC projects, as well as molecular signatures of the LINKS projects. The homepage also provides links to the Global Gene Drug Network. Our four main modules, which include Query a Gene, Query a Gene List, Query a Known Drug, and Query a New Drug. We also provide a link to our help pages. For the global network, the module allows a user to set a cutoff on gene-to-drug correlation, drug-to-drug correlation, as well as gene-to-gene correlation. The default settings are the cutoffs that we used in our manuscript based on network analysis. After we hit submit, our tool generates a gene-to-drug correlation network. Each circle represents a gene knockout. Each diamond represents a drug treatment. A positive correlation indicates similar inhibitory effects induced by a gene knockout and a drug treatment. The network contained clusters of drugs that were designed to inhibit important oncogenic and cancer progression pathways, such as the MEK inhibitors with genes involved in the BRAF and MAPK pathways. MDM inhibitors form a cluster with many DNA repair genes that govern cell apoptosis, such as TP53, CHEK2, ATM, and MDM2. We also observed a cluster of inhibitors of the ERBB family targeting the EGFR, ERBB2, and ERBB3 genes. Genes involved in the mitochondrial respiratory chain formed a very dense cluster that included the COX genes and the NADH enzymes. After we checked the results of the global gene drug network, the main modules of our tool allow users to either query genes or query drugs. For query genes, a user can search for drug treatments that achieve similar effects as the knockout of a gene, or a drug that can simultaneously up or downregulate a group of genes of interest. For query drugs, the tool searches for gene knockouts that achieve similar effects as the treatment of a known drug. It also allows the user to analyze a new investigational compound based on the SMILES codes, which is the chemical formula of a compound. As a use case of DepLink, here we further investigated CDK6, which is one of the most promising targets of cell cycle-based cancer therapy. For presentation purpose, here we summarized results from Module 1, Query a Gene, and Module 3, Query a Drug. CDK6 was found to be an essential gene in roughly 10% of pan-cancer cell lines assayed by the broad DepMap. In particular, viability of myeloma, leukemia, and neuroblastoma cells was strongly dependent on CDK6. Here are the top 10 drugs that achieved the highest correlations to CDK6 knockout. The drugs formed two clusters. The upper cluster included two CDK4 and CDK6 inhibitors, palbociclib and ribociclib, as well as ponatinib, which promotes G1 cell cycle arrest and synergizes with palbociclib. The lower cluster of drugs was composed of 7 MEK inhibitors. The 7 MEK inhibitors form an interconnected network. Our data aligned well with previous reports showing potential synergistic effects between CDK and MEK inhibitors, which we further discussed in our manuscript. With our tool, we also provide detailed user manual and help pages, as shown in these screenshots of all the 4 main modules, as well as the global gene drug network. In summary, the proposed tool is, as far as we know, the first software that allows the broad biomedical community to navigate, visualize, and integrate genetic and pharmacologic dependency maps together with molecular signatures of cancer. We expect DepLink to continuously evolve with rapidly growing high-throughput screening data resources. Our tool is now freely available for academic uses at this link. I would like to thank all the authors for their tremendous contribution to this project, and the Center for Research Computing at the University of Pittsburgh for hosting the RShiny app on a high-performance computer. We would also like to thank all our funding resources. This study is not possible without their generous support. Thank you for watching this video. Please do not hesitate to contact us if you have any questions regarding our tool or this study. Wish you all enjoy the GLBIO conference, either in person here at Montreal or virtually. Thank you.